Welcome friends. In the first part of this lecture series, we talked about the introduction to proteins and why there is a need to study proteins. If you haven't watched that video, I suggest you to have a look on that first, then come here. There is a link in the description box. This video is about the amino acids. Okay, so we start with a question here, that is how proteins are formed. The synthesis of proteins is by amino acids. Amino acids are used for the synthesis of proteins. These amino acids, they act as building blocks for proteins, like the bricks are used to make a house. But the question here is how we join one amino acid to the another amino acid, like we use cement to attach one brick to the another brick. The simple answer to this question is peptide bond. Peptide bond is used for the attachment of one amino acid to the another amino acid. But we are not going into the detail of the peptide bond formation here. That we would discuss in a separate video. Let's draw a diagram for what we have studied so far. In the diagram you can see different colored boxes. These colored boxes actually are representing different amino acids. So these amino acids, they are attached with each other with this black thread. Okay, so this black thread is nothing but the peptide bond. Let's go to the next slide now. What is an amino acid? This is the general structure for amino acid. The amino acid is an organic compound which is formed of the following constituents. So now we are going to know about the chemistry of amino acid. What we are going to find in an amino acid structure, there would be a central carbon atom to which there would be some groups of atoms which would be linked to the central carbon atom. The first one is the amino group. You can see in the diagram, the left side of the figure, the NH2 group. The B is the carboxylic acid group, COOH on the right side. Then we have the H atom. And then the most important thing is the distinctive R group, which is also called as side chain. So we have to focus on this R group because this one particular group is going to differentiate one amino acid from another amino acid. This group is responsible for the identification of particular amino acid. So what I am trying to say is that in every amino acid, the first three groups, that is the A, B, C, these would be same. And the fourth group, which is the R group, it would be different in all amino acids. But there is an exception to this general structure. That is, it is common to all, but proline here is an exception. Proline is an amino acid whose structure is not resembling to this general structure of all these amino acids. We will talk about this concept later when we will discuss the each and every amino acid in detail under the topic of classification of amino acids. Now I want to ask you something. Did you notice a special thing here? In this figure, the four groups are different. We know the valency of carbon is 4. That means it can attach 4 atoms or the group of atoms to it. But in this case, all the atoms or the group of atoms which are attached to the central carbon atom, they are different. NH2 is different, this hydrogen is different, and the third group, which is the carboxylic acid group, this is different. And we know about the R group, which is different in all the amino acids. So 4 groups are different. So the question here is, if all the 4 groups which are attached to the carbon atom are different, what we are going to get? The result is chirality. Now what is this? About chirality you can find more in a separate video about which I have mentioned a link in the description box. You can check that. For now, we can say the presence of different atoms or the group of atoms results in the asymmetric carbon. This asymmetric carbon is nothing but which is attached to 4 different atoms. So when this asymmetry in the groups which are attached to the carbon is achieved, a class of the stereoisomers is produced, which are known as enantiomers. So what are these terms, that is the stereoisomers and the enantiomers? Let's have a look on that. This is the figure related to enantiomers. First we talk about the stereoisomers. These are the different forms of the same compound. So there are more than two compounds, there can be two or more than two compounds which have the same molecular formula, but they would be differing in the 3D or the three-dimensional orientation of their atoms in space, or we can say the arrangement of their atoms would be different in space. Now we talk about the enantiomers. These enantiomers, they are a type of stereoisomers, a class of stereoisomers. What specific points these enantiomers have? They are actually mirror images of each other. In this diagram you can see a dotted line is between these two molecules, which is representing a mirror here. So they are mirror images of each other. Okay, and also one important thing is here, that is they are non-superimposable, that is they cannot be superimposed on one another. That or we can say we simply cannot put one molecule on the top of the other molecule. The simple real life example of this property is our hands. Our hands are the mirror images of each other. In this diagram you can see here, if we take the right side molecule and put this molecule here, we can observe that the NH2 group would come here and the H atom would be here. In this way they are not anymore same. So this was all about the introduction to amino acids. In the next video we would be discussing about why there is a need to study amino acids in detail and not just remembering their names. Thank you.